All right. So when you're starting with aerobic gram-positive bacilli, I was making a joke to Lauren earlier today, saying that when I was looking at these two PowerPoints, they're awful. I was like, I must have been drinking or something because half of them don't have the information that you need on the PowerPoint. So it's written up here on the board, all right? So one of the things that we're going to have to know, is the virulence factors for Listeria, okay? Listeria. So there's 5 that you're going to have to know and know the differences between the 5, okay? So catalase and being catalase positive, that's actually considered a virulence factor for Listeria. Then the big one is this guy right here. There's a protein called P60, which induces phagocytosis. This is really big when you're talking about Listeria, the P60 protein. Okay? Internalins, I think, surface proteins that are used to invade the actual mammalian cells. Okay? Listeria lysine O is a hemolysin. So if it's a hemolysin, what's it going to do?
Lyse red blood cells.
Yeah, lyse red blood cells. Okay? And then your last one is just basically a calcium-dependent adhesion. So what I would like you guys to know, if I ask you, like, what are the virulence factors, you'd be able to give me a list and basically give me a little description about what each of them do.
I have a question there.
Yes, ma'am.
On the PowerPoint, there's one called Act A.
I'm glad you asked about that. The Act A, you can cross that off of your PowerPoint. Don't worry about that.
Okay.
It's not important for our purposes right now, and I'm glad you asked about that. I think it's the very last one, maybe, Act A. I think. Yeah, it is. So just cross that off of the PowerPoint. Are you there, Mercer? Are you on the right?
Are we on meningitis or
No, not on meningitis.
is it actomyositis?
It's just, I think, called Act A, right?
Yeah.
Go to the Listeria PowerPoint and you'll see under there, there'll be a list of these guys and then it'll say Act A and just cross that out. You don't need to know it. Okay, so that won't be on the exam. Okay? All right. Test-wise, that goes back to Listeria. Okay? Piperate hydrolysis positive, which that should sound familiar to you, that test. Okay? If it's not sounding familiar to you, you're in trouble. There was a specific organism that we talked about that actually used that test a little while ago. Bioescalin positive, that should also sound familiar to you, okay? And then the CAMP test positive. All of these should sound familiar, okay? But all of these are positive for Listeria, okay? All right. Then the other thing I wanted to talk about that wasn't listed very good in the PowerPoints and that you needed a little bit more information on. Well, first of all, are you guys good with Listeria, or do you have any questions about Listeria? I mean, we're going to talk about it in the PowerPoint, but I want to make sure on this stuff, are you guys okay? Okay. All right. Bacillus anthracis. Okay. Your 3 big virulence factors are listed right here. Your PA, EF, LF. So you have a protective antigen, edema factor, and lethal factor. Okay, those are going to be your big 3 when you're talking about anthracis. Now, I talk about subtilis and serious a little bit, but this is specific to anthracis, okay? The other thing to know about this is that you have the 3 modes of ingestion, basically. So you have cutaneous, inhalation, and gastrointestinal. Now, with the cutaneous, which is the most common, you're going to have some kind of black central area that would be on the lesion, and usually the infection will remain localized, usually. Okay, sometimes it can go systemic, but not all the time. Usually these infections are fairly common, especially like in Russia, where you're working with cattle and that type of thing. The cutaneous isn't that big of a deal. It's the gastrointestinal is a big deal, and so is the inhalation is a big deal. Okay? All right. The other thing is they're actually characterized on a BAP as having whatever is called a medusa head. Okay? Kind of swirling all over the place, I guess. But that's usually what they'll refer to that in a lab. They'll actually refer to it as a medusa head, and that's characteristic of anthracis on a VAP. Okay? All right. The other thing for anthracis, you're talking still about being catalase positive. Here we go again. They're all getting to be catalase positive. Non-modal. This is very important because this distinguishes it from the other species of Bacillus. Okay? It also grows in high salt content, usually around 7%. And some of the time with this one, it's susceptible to penicillin. Okay, so this is also kind of another one that it's sort of distinguishes it from cirrus. Okay? The non-modal part is probably the most important thing on that list. Okay? Now, your last little thing, and this is like jumping ahead to the next section, would be Neisseria. And when we're talking about Neisseria meningitis, obviously we're talking about meningitis. So the serotypes that we're talking about are going to be A, B, C, Y, and W135. And these are most often associated with epidemics. I'm actually going to go through these in the lecture. Each one is associated with something a little bit different than the other. And you'll have to know those differences, okay? But that will be in the lecture. Current vaccine. So on your slide where it's talking about current vaccine, make sure you fix this, okay? Because the slide wasn't updated. Current vaccine right now, they have it for A, C, Y, and W135. However, they started with a B back in 2014, and this was suggested for 10 to 25-year-olds. Okay? But before, they didn't have the serotype B in the actual vaccine. Okay? All right. Any questions on the Neisseria meningitis? So, Listeria. Associated with the spectrum of clinical syndromes. Okay? Have you guys heard about this one before?
Yeah.
Pretty much. You're saying no, but who's saying yes? Who's it usually affecting?
I'm thinking mono.
What were you thinking?
Is it mono?
What? I didn't understand what you said.
It's not. I'm thinking something else.
It's not mono.
No, because
So
that's
I didn't understand what you said.
It's wrong. What
What
I
were you
said.
saying?
Oh, pregnant women, immunocompromised people, older people.
Pregnant women usually is like your go-to, where they're like, don't eat hot dogs during your pregnancy
Yeah.
because it's processed food. Yeah, it's processed food. So look at that. The most common route of indigestion is contaminated food, and usually it's processed food or raw milk, cheese, and poultry. Okay, so what happens is the mom gets infected, and then that can cause a stillbirth or what am I trying to say here? Spontaneous abortions. But then if it gets passed to the neonates, you have an early onset or a late onset. So differences between the two, obviously one's early, one's late, okay? But what is the symptoms of them? Early, you're looking at that they might be associated with aspiration of the infected amniotic fluid, and so they would be treated on antibiotics. The late onset is the one that's a little bit scary because that actually happens 7 days to a couple weeks after the birth, and usually that manifests itself as meningitis. Okay, so early versus late. There was your list of virulence factors. Remember, you can cross the last one out. That's Act A. You don't need to worry about that one. But remember to get P60 in there and catalase positive as well. Okay? Let's see. Also, these things can be hemolytic on VAPs. You can put them on. The non-hemolytic strands are found to be not virulent with no intracellular spreading, which is kind of interesting. So those specific strands basically aren't doing much in the world of chaos. Okay, isolation from clinical specimen. So you can get it from blood, CSF, genital tract specimens, amniotic fluid, and biopsy specimens. Direct inoculation and incubation will usually heal the organism. The thing about this one, it has kind of a funky incubation and temperature scale that goes along with it. Isolation from foods are going to involve broth enrichment and subculture, so they're not very straightforward. So when you're looking at lab, there's a picture of it, sorry, I'm jumping ahead. When you're looking at lab diagnosis, you're looking at Bacillus, okay? It's going to grow well on BAP. Here's what's weird, is that it does grow at 4 degrees Celsius, and that's a major factor, because this is going to help you differentiate between some of the other organisms that we were talking about, that we will be talking about, okay? The optimal growth is 30 to 35 degrees Celsius, but it has a weird range. It can go from like 0.5 to 45 degrees Celsius, but sometimes they'll do this thing called cold enrichment, where they actually grow it at 4 degrees Celsius, and it will take a couple weeks for it to grow to make sure that that's what they're dealing with, which isn't very good because it's taking a couple weeks in order to make the diagnosis, right? Also, catalase positive, and it's beta hemolytic. Treatment, fatal if left untreated. However, it's susceptible to a variety of antibiotics. I would look and probably focus on the two that it's not active against, because that would be an easy question. And patients should be treated for at least 3 weeks. It's kind of a long treatment. Okay? And the other thing, you guys, to remember is that the test, the biochemical test that it was positive for, which is written on the board, piperate hydrolysis, starting with that one. Okay? So that's Listeria. Good God, I just forgot how to pronounce this again. Erysipelothrix rhusiopathiae, I think. Did I say that right? We were actually in the office, like, listening to people pronounce this, because I was like, how the hell do you say this? Okay. Gram-positive, this is catalase negative. Catalase negative, okay? Pleomorphic rod, it has long filaments, which is usually a telltale sign when you're looking at this, the long filaments. Human occupational exposure. So individuals who work with handling fish and animal products are most likely the ones to end up getting this. It survives in environmental sources, produces diseases. These are the following the diseases that it produces. But the other thing is, where you're looking at Listeria, this one lacks the ability to grow at 4 degrees Celsius. So there goes back to the 4 degrees Celsius. Okay? This would be the major thing. What else is different about the Listeria versus this one? Catalase negative, right. And quite frankly, what about the shape? Versus what? Versus the filaments, right? Okay. So picture A is actually some strep rods, and picture B is this last organism that I was talking about. You can see there's like a really long one in the middle that almost is forming a backwards L. They're long filaments like that, but usually they're even a lot more funkier. This isn't like that great of a picture, but just so you can see like even the size difference with some of them. Okay, clinical significance, mild localized cutaneous infections. Let's see. You can get acute bacterial infections from the traumatized skin, which can disseminate to other organs. That is possible as well. Okay, that would be your systemic infection. Yeah. So that all I'm saying there is that it can go, besides the initial skin infection, it can travel to other organs. Okay? Corynebacteria. Okay, large diverse group of bacteria. These include a whole heck of a lot of different things. Okay? One of the things to remember is that these species are found as normal biota on the skin and mucous membranes. That's pretty significant. Medically significant pathogens are catalase positive, and they're going to be non-modal. So non-modal wise, who does that not help you with? If I'm asking non-modal, who is it not going to help you with? Bacillus and Moraxella, right? But there again, would Bacillus and Moraxella be normal biota on the skin of mucous membrane? No. So that's like an easy one right there. Okay? There's actually 40 that are clinically significant, which is a lot for this group. They're lipophilic, so they're tending to combine or dissolve with fats. They're pretty fastidious and grow in standard culture media. And actually, they sort of look club-shaped when you look at them under a microscope. They have this club-shaped appearance, okay? But these can be hard to identify. All right, diphtheria. We've all heard of C. diff, right? So we have the toxin. You're talking respiratory. Incubation is 2 to 5 days. The illness begins gradually with the tonsils, which would be the most common infection, okay? And then we also have cutaneous, which I don't know if you guys knew about that, which would be unhealing ulcers, is usually how it takes form. Did you know, did anybody know that it did that, besides the respiratory part of this? Highly pleomorphic, okay, so it can take many different shapes. Let's see. Optimal growth is 37 degrees, so it would be the same as what we're growing our stuff in. However, it has a 15 to 40 degree range. Okay? 15 degrees Celsius to 40 degrees Celsius range that it can grow in. So it's pretty flexible in terms of the temperature. Okay, but optimally it likes 37 degrees. Complex growth media requirements. So media-wise, you plate it on something called CTBA media. CTBA. Okay? Now, this was diphtheria. Now we're going to talk about 2 other ones. Jejunum is the next one. Where people usually get messed up is between jejunum and urolithium. Okay, that's usually, people get those confused, but there's like a really just simple way of remembering. Urolithium, think of urine. Okay, UTIs, urine. Okay, jejunum really isn't going to do that. This is going to be part of normal skin flora. Okay, usually it goes after patients that are immunocompromised. Of course, again, the patients that have catheters or prosthetic devices or ports. The range of infections are very wide. So septicemia, meningitis, joint infections. Yes. The other thing is you can get skin complications from this bug as well, meaning, wounds that just won't heal. The other thing I wanted to point out is that this guy is urease positive, and it reduces nitrate. Urease positive and reduces nitrate. And, unfortunately, it's resistant to a wide range of antimicrobials. So that one's not looking too good. I go laughing. It's not looking too good. All right. It's been a long afternoon. What can I tell you?
She's not a lot of coffee.
I have. My eyes are like bugging out of my head right now. All right. Urolithium. So think of UTI or urinary pathogen, okay, which they're primarily described as a urinary pathogen. Okay? This would be one of the most frequently isolated and clinically significant strains. Now, rapid urease positive tests, non-hemolytic white colonies, does not ferment glucose. These are all important things to remember. Okay? All right, moving on. Okay, one thing I wanted to point out to you guys, on your, and actually Lauren found this, not me, on your study guide that had the organisms listed for exam 2, A. hemolyticum was not listed. You might want to add that to your list if you need it on that list. Okay? That's like the big list that has like the descriptions of the organisms. Okay? Add that one to it. It wasn't on there. All right. So this species, you're looking at 3 medically important species, but we're only going to get into really one, which is Hemolyticum. Facultative gram-positive bacilli, it can be slightly curved, also catalase negative, okay? And Hemolyticum is the one that usually is on everybody's to-go list, okay? So with Hemolyticum, all right, just in general, many of the rods are pleomorphic and they're branching. okay? This would distinguish them from group A strep, the morphology would, okay? The problem with this one is that it's going to produce small colonies on BAP with black opaque dots, which may or may not be observed, but your hemolysis might be the same as group A strep. That's where you can get into a little bit of trouble, and if you can see what it does clinically, you're also in trouble because group A strep can cause soft tissue infections, right? So you got to pay close attention to what you're doing with this. The other thing is it's lipase positive and it's penicillin resistant, okay? Erythromycin would be the drug of choice for this guy. All right? So just remember when you're studying, look at this and then look at group A strep to see if you can find out some differences between the two. See, how are you guys feeling? Is all this like kind of blurring together, unfortunately? Or are you doing okay? I'm trying to gauge you. What's going on? Is it?
Yeah.
It's too soon.
Everything is awesome. Gardnerella vaginalis. Yay. Short pleomorphic gram-positive rod that often stains gram-variable. Now, why do I have gram-positive if it stains gram-variable? Does anybody know?
It didn't say
Oh, it does say?
Yeah.
Damn it. I didn't even notice that. Gram-positive cell wall type. So what's your gram-positive cell wall type?
Thick peptidoglycan.
Think, what else do you have that's going on
Cyclic
there?
acid.
Yeah. Okay, good. Normal biota in the UI tract, okay? But this one causes BV, which is not good. It can be isolated from as many as 40% of women without BV symptoms. And here's the problem. This is actually going to be, BV would be a direct result from a reduction in lactobacillus, which is normal biota in the vagina. In that population in the vagina, it would rise the pH instead of lower the pH, okay? And that's where the infection comes into play. On this one, you're going to grow best at a higher CO2 level, about 5 to 7%, and it's still going to be about 35 to 37 degrees Celsius in terms of optimal growth, okay? The other thing that you guys need to know is that it's going to be best plated on human blood bilayer tween auger, or HBT auger. Can you just say that again?
Human blood.
Human blood bilayer tween, HBT auger. And it's on the next slide, after you guys wrote it down. It's on there. I'm in this weird mood today. HBT. Okay. Susceptible to pretty much a not so bad variety of choice of drugs right there. Okay? The big thing to remember is that it's going to be gram variable. Okay? But actually, when all is said and done, it's really classified as gram positive. Okay? So that one's causing BV. Alright, cross lactobacillus out. We're not going to have that on the test. Okay? So that slide is out. Clinical significance is out. Isolation and identification, out. Okay, now we're getting into Nocardia. Alright, found worldwide in soil and on plant material. It can cause pulmonary and cutaneous infections. This one, I think you guys are going to have to be a little bit careful with Nocardia and with Bacillus and Moraxella. Okay? Look at that. 40% of diagnoses are made on an autopsy. Kind of creepy. Depending on if it's inhalation, really. Okay? But it's associated with impaired immune defenses, number one. The other thing, too, is that it's going to grow in non-selective media, 22 to 37 degrees Celsius. Long branching filaments, and they're going to be partially acid-fast stained. That's going to be your best way of looking at those, is partial acid-fast stain. And then treatment is going to involve drainage of the wound and antimicrobials. Okay? but people who survive often suffer significant tissue damage. All right, Whipple disease. So this is the agent of Whipple disease. Anybody know what Whipple disease is? It usually affects like middle-aged men. Have you ever heard of this one? Yeah, dude, you have something to look forward to.
Well, we don't have the vaginosis one, so.
I know, right? That's actually a good one. That's good. But if our pHs are correct, we won't either. So, you know. Symptoms for this one, basically, it's a lot of abdominal symptoms. So you have diarrhea, weight loss, abdominal pain. You can have neurological dysfunction, sensory changes often occur. And like I said, usually it's more common in middle-aged men. And sulfa treatments are usually for 1 year. Okay? So that is a long time. So that's Whipple disease. Well, I think the problem is with this one is that it goes undiagnosed for such a long time. It's not like one of your go-tos that they're like, oh, Whipple disease.
It's kind of like it's intracellular, like
The other one's
Rickettsia,
yeah.
Call them Whipples
or
instead of
the other one
Whipples.
that we covered just
Listeria.
Listeria.
Listeria,
And
yeah.
that one needed a long antibiotic.
Yeah, long antibiotics for that one. All right, Bacillus. What I want you guys to pay attention to here, which is interesting, is that it's classified gram-positive. However, it's gram-variable, which we've been noticing in this lab a lot. Gram-variable, okay? So aerobic or facultative anaerobic bacilli, and they're going to form endospores. They're going to be in the soil and environment as well. Metabolically diverse. Some are going to grow as high as 55 degrees Celsius. So anthracis, I wrote that on the board, susceptible to penicillin, non-nodal, and non-hemolytic. Okay? Serous, this is a cause of food poisoning. I don't think I have anything else about that one that's big to remember. Usually they had it where it was actually caused from fried rice. I don't know if you guys remember that from 2010. Did I have that on there? Okay. Yeah, fried rice syndrome is what they used to call it for Bacillus cereus. And this one you guys work with in the lab, okay? But cereus would be nodal, okay? There's a picture. All right, last little thing. There's your points to remember. Remember, here's your big one again about the 4 degrees Celsius. They keep saying this over and over and over. So if you guys get it wrong, it's not going to be a good day. These guys, general characteristics you can see right here, non-nodal, non-spore forming, cytochrome oxidase, that's what I just did for you right now, is when it turns purple. They're going to be catalase positive again, and gram-negative diplococci that basically look like coffee beans, like I said before. They're cafenophilic. What does that mean again?
CO2.
Higher CO2, okay? Let's see. Primary human pathogens in this species are gonorrhea and meningitis. They're both going to require iron for growth, and they're going to compete with the human host by binding transferrin to their specific surface receptors. The other thing I wanted to say was that gonorrhea is always pathogenic and not considered normal biota, but meningitis can actually be found as a habitant of the upper respiratory tract, okay? So that's the difference between the two right there. Obviously, you're going to know the difference, but that's one of the things that's interesting about both of them, okay? All right, so for gonorrhea, you have a whole mess of virulence factors going on here, okay? Let's see. The immunoglobulin A, okay, that's going to cleave the IgA off of mucosal surfaces. And obviously, for gonorrhea, it's going to have to go through a lot of mucosal surfaces to actually attach and to get to where it needs to go. This one, as you saw, it's going to require the use of chocolate agar. And again, the CO2 level is going to have to be a little bit higher than normal. Okay? Function-wise, you don't really need to know, like, what is the you know what the capsule does, but you don't need to know like specific cell membrane proteins or anything like that. You know what endotoxins do already. And competing for the iron, of course, would be the very first one. So basically, this is sort of self-explanatory. Okay? I don't think you guys need much help on this one. So sexual contact, primary reservoir is the asymptomatic carrier, which is kind of scary. It's a nationally reported disease. Anybody have any ideas about the neonatorium? If I said that right, I don't know if I said that right. Ophthalmia.
It's the number one cause of blindness
Okay.
for
Yeah.
infants or
For
people.
infants,
Or,
and usually what they do when the mother's actually giving birth is they will put in silver nitrate in the baby's eyes to actually avoid this. What? No, this is for gonorrhea. Yeah, silver nitrate in the eyes for that. Yeah, chocolate agar at room temperature is usually the best way to go, and then I was talking about the incubation. The other thing that I wanted you guys to know was that it's going to depend the virulence on if or if it doesn't have pili. Okay, so there's T1 through T5 is based on the presence or absence of pili. So T1 and T2 possess pili and they're virulent, whereas T3 through T5 they're absent, so they're avirulent. Okay?
Alright, so it's T1 through 5.
Yeah, T1 and T2 they have the pili, and the rest of them T3 through T5 they're absent. So they don't cause infection because they don't have anywhere to attach to because the pili is absent. You got it?
Mm-hmm.
Okay. The other thing is incubation can be for men 2 to 7 days and cause acute urethritis. 95% are going to show acute infections. Women, it's going to affect the endocervix. 50% of the cases may be asymptomatic. And that's how it's going to get carried from place to place to place. Rayon swabs are preferred because standard swabs will actually dry out the specimen. And we also need a transport system. Usually it's amines. They usually need to be plated within 6 hours. And if you go on chocolate agar, there is a specific agar for gonorrhea. It's called NYC or New York City agar. Okay? And again, oxidase positive. Okay, meningitis. This slide is just a hot mess, okay? So use what I put up on the board up there, all right? Note, remember that it can be an upper respiratory tract of people, but that A, B, C, Y, and W135 are most often associated with epidemics. Now, if you break down those numbers, right, group A is associated with pandemics. B and C are found most likely in the United States, with B being community acquired. Y is meningococcal pneumonia, and W135 is invasive disease. Okay? So classical symptoms, I mean, this can be spread through oral secretions and droplets. I mean, the big thing is the frontal headache and stiff neck. And then here or there, sometimes the fever. And then sometimes the person actually will get a rash. And that's usually the go-to thing that, we might be looking at meningitis here. And if it's not treated, the fatality rate is 10 to 15%. So it's very serious that you get in and get checked out. And when I'm talking headache, I don't mean like, oh my God, I got a migraine. I mean headache, like you can't stand to be in the light. You got to be in a dark room. Your head is pounding, type of thing. Okay. All right, and this one's still oxidase positive. The other thing for meningitis is they have latex agglutination tests that are available as kits to detect group-specific surface antigens. So that's one way in the lab that they can detect Neisseria meningitis. It's almost reminded me of like Staph aureus, kind of, the latex agglutination, but in a different type of way. Okay? And there's a picture of the meningitis going to Bacilli. Right, this is just a little blurb about the vaccine. You don't need to know this slide for the actual exam, but this is just a blurb for you to read about the vaccine itself. All right, Moraxella, opportunistic pathogen. Okay, so this is not normal flora. Recognized as a cause of upper respiratory tract infections and lower tract infections in adults. especially those with chronic obstructive pulmonary disease. Advanced age, immunodeficiency, neutropenia, and chronic debilitating diseases are often a result where you can pick up Moraxella. And this would be the third most common cause of ear infections and sinuses in children, okay? And we actually have, I think, a Moraxella sample in the lab, and we definitely have the laboratory diagnostics to confirm that it's Moraxella. So maybe you guys will do that next week, okay? All right, unlike Neisseria, this one can grow well at 28 degrees Celsius, okay? But here again, intracellular gram-negative diplococci has an elevated center, but the good news is that it's usually inhibited on the gonococcal selective augurs, such as the NYC auger that I was talking about, the New York City auger, okay? So, and it can tolerate lower temperatures as well. But 28 degrees for this one.